Thank you very much, everyone. My name is Opiyo. I'm from Ohio State University, and my topic today is application of artificial intelligence in plant pathogen effectors. So I'm going to start with this. I have a bowl of tomatoes here, and these are healthy tomatoes. And then I have another bowl of tomatoes. These are sick tomatoes. So when you go to the market, you always want to get this type of tomatoes, but not this type of tomatoes. So the question is, how do you move from here to here? So this is what I'm going to talk about. So you move from here to here because of infection by bacteria. So this bacteria delivers what's called effectors. So these effectors, this is kind of the tomato cells, and these effectors, when delivered by this bacteria, it affects this bacteria, and from there, it becomes rotten. And these effectors are also found in animals and also human. For example, when you eat maybe raw vegetables and you get food poisoning from salmonella, it's the same principle. Sorry. What salmonella does, it delivers those protein in your system and then you get sick. Okay? So what are these effectors? So these are proteins that are secreted by pathogens. It can be bacteria, it can be fungi, or viruses. And what it does, it goes and it disorganizes your immune system, either in plants or in animal. And this is an example of effector protein. So it's basically all these pathogens have these effectors. So what they do is they deliver them in your system so that you get sick. Okay? So how do you discover these effectors? So in bioinformatics, when a pathogen is discovered, it has to be sequenced. For example, when we got COVID-19 virus, COVID-2, when people started getting sick, they didn't know what the virus was. They had to sequence the virus. And then after sequencing the virus, they had to go and look for what type of virus it is. So there are methods for finding these sequences. One of them is called basic alignment search tool. What happens is, assume this is our protein that we don't know what it is. So you have these databases that have other proteins. So what it does, this protein is sent through databases and there's payloads similar to such. And then the ones that are very close to this protein of our interest will be on top, and then in further, that's your protein. Okay. So sometimes, if you sequence such a protein, you may fail to get the homolog in the database using this method. Then you go to another conservative method, which is called Position Iterative Specific Blast. With this method, the first iteration is you do the pairwise and then you align them together. Then you build this position specific scoring matrix, and then you now get the protein. This is much better because now you're using the alignment. So if you had failed to get the same protein during the previous method, you can get a similar protein now in this method. Okay? So if you would use this method again and you fail to get the protein, you can go to the next method, which is called profile hidden Markov model. This is where it's a little bit better because now you're building alignment from all these proteins, and then I'm using the profile to search your protein. Okay, so these are the most common methods that people use to identify some of this protein. Okay, but these methods have problems. So they work very well when your proteins are very similar. So for blast, you need around 40% identity. For cell blast, you need around 40% identity. And for hidden knockoff model, we need around 25% identity. But most of these effectors are below 10% identity in what we call twilight zone. This is now where the AI comes in. So with the AI, you don't need to align it. So how do you use AI to do that? Okay. AI, you don't have to align them, but one thing you can do is can find a composition of each amino acid. For example, in this protein 1, how many times does this M appears? That is the composition. And then the protein 2. So instead of aligning them, now use that composition. Okay. Another method that you can use is you can use what they call the pipeline. Is how many times do these amino acids appear together? Okay. So instead of aligning them, you can now use these two for your AI. And the last variable you can use is the physicochemical properties, because all these amino acids have their physicochemical properties. They have their mass, their volume, and so forth. So you can use that. So now you have your variables from those proteins. Now you can use to do your mining. Okay. Now, since you've now converted all this into that chemical properties, you can now build models. After building models, you can now use these effectors. So now, if you sequence and find new proteins, you can now go in your models and identify the novel effectors. Okay. So now I'm going to show you where we had a symptom that was similar to tomatoes. So when they went and did the sequencing, they used the other previous method that I mentioned, that they could not find any effectors. But when they used the AI, they were able to find the effectors. Okay. So this was the protein effector that was used for trading, and they were using databases, using their physicochemical properties. But using alignment, they could not find these effectors from omicities. But using AI, they were able to find these effectors because these effectors was also causing the tomato to rot, but they were not yet discovered. Okay? So, this is how protein works in biology. Protein work by using 3D structure. Even these effectors, when they're delivered in organism, they use the 3D structure. So what we did was try to find out if these proteins were similar structurally. So this is the structure of the big protein, and this structure of the small protein. But you can see structurally they're similar, they overlap. So what we hypothesize is that when this is delivered in the tomato cells, it goes and does a similar thing with what this one is doing by binding to tomato cell. So this big protein, that is only this part here. So what this part is functioning is similar to this part here. Okay. So all this is now being done computationally. But when you work with computers, you have to work with people in the lab to prove that what you are trying to identify or model is really true. Okay? So this is now the lab experiment that we did. So this is a tomato leaf. So when these organisms, so this one is the ones that have the protein. So when they attach tomatoes, you can see that you have all these pestles which are kind of red. So we counted them. So if you count them per leaf, So this one have that protein in them. You can see that it has a lot of vessels. And this one here is the mutant, that means they've removed this protein. You can see that it's not possible for it to affect all the tomatoes. So what we did was we replaced this new protein that we discovered, and you can see that at least it affected the tomatoes. And also here now, we put back that protein that was removed from here. You can see also that it did what? It affected the tomatoes. Okay? So basically what we discovered using AI, it was able to go and identify those proteins that were able to identify using the previous methods. For example, if when the COVID-19 virus was discovered in China and it had no identity from the database, we would not know what type of virus it belonged to. But we're lucky that the other previous methods that I showed before were able to identify them. But sometimes you have some of these pathogens, either in human or in plants, that the other methods that do not use AI cannot be able to identify them. That's why the AI comes in, okay? Okay, so this is another example that we use, we can use effectors. So this is a disease of wheat. So this one affects wheat from fungi. It destroys wheat in the field, and then also when the wheat is harvested, it can also get into the flour. So it becomes poisonous to human being. So if you use effectors, you can use bioinformatics and try to silence the genes that are vulnerable to the effectors, and then you can also use the other genes. And using bioinformatics, you can now develop resistant wheat. So using AI, you can be able to control this pathogen in wheat. With that, I thank you very much, and if you have any questions, thank you.
Got a couple minutes if anyone has any questions.
Yes. Any question? You are right, but all of them are effectors. So they are basically affecting the same system. Yes. Yes, yes. Yes, because these effectors sometimes they interact with those genes from the host for them to compromise the immune system. So if you can silence those genes from the host, assume they are not housekeeping gene, that might not help.